So I want to transport you back to the year 1969.
Oh, right. The Li-Fraumeni discovery.
Exactly, yeah. So two researchers, Frederick Paley and Joseph Fraumeni Jr., they are poring over just thousands of medical records.
And they notice a pattern that is, frankly, devastating.
Right. They are tracking these families where cancer isn't just, a random stroke of bad luck or like a product of environmental exposure.
Right. It's a terrifying inherited certainty for them.
Yeah. They begin documenting these families facing a staggeringly high cancer risk. I mean, we're talking about 50% of carriers developing malignancies before the age of 40.
Which is just unheard of.
It is, and a lifetime risk exceeding 90%.
Yeah.
So today, the medical community knows this as Li-Fraumeni syndrome, or LFS.
And honestly, it remains one of the most monumental discoveries in modern medical genetics.
Absolutely.
Because as genomic sequencing caught up with clinical observation, we eventually we pinpointed the exact driver of this syndrome.
Which is a mutation in a very specific gene.
Right, the TP53 gene.
The famous guardian of the genome, as they call it.
Yes, exactly.
Right.
So when TP53 is functioning correctly, it acts like a cellular emergency brake. It stops damaged or mutating cells from dividing until they can be, well, repaired or destroyed.
But in LFS, that brake pedal is fundamentally broken right from birth.
Right. And what is particularly striking here is how this broken brake pedal actually manifests in the clinic.
Right, because these patients are susceptible to various cancers, but there is this one hallmark tumor they are just exceptionally prone to.
Yeah, osteosarcoma. It's a highly aggressive, bone-destroying cancer.
Which is usually incredibly rare.
Very rare. Maybe 3 or 4 cases per million people in the general population.
Wow.
But if you have Li-Fraumeni syndrome, your risk is hundreds times higher.
And that brings us to the central mystery of our deep dive today, because just having a break in TP53 gene tells us the brakes are cut, sure.
Right, but it doesn't explain why the car specifically crashes into the bone, so to speak.
Exactly. It doesn't explain how that generalized genomic vulnerability translates into this very specific hyperaggressive bone tumor.
And that's what we're looking at today.
Yeah, so today we are tearing into a massive multi-omics study. The mission here is to hunt down that missing link.
The exact genetic bridge between this inherited curse and osteosarcoma.
So, okay, let's unpack this, because you're here to learn quickly but thoroughly without getting overwhelmed. So we are going to break down every mechanism step by step.
Right, and to find that missing link, the researchers, they couldn't just look at a handful of genes.
They had to cast an incredibly wide net.
Exactly, across the entire transcriptome.
So they're looking at all the RNA messages being sent out by the genome at any given moment.
Yeah, and they utilized a technique called WGCNA, which is weighted gene co-expression network analysis.
I want to pause on WGCNA for a second, actually, because it is a brilliant approach.
It really is.
Because instead of just looking at a list of genes that are active, it looks at the network architecture, right?
Right. It figures out which genes are constantly talking to each other, and it groups them into these functional modules.
Like finding the cliques in a high school cafeteria.
Yeah, that's a good way to put it. It groups the loudest, most coordinated conversations in the genome.
Okay, so by applying this to LFS patient data, they identified a pool of 1,410 genes intrinsically tied to the syndrome.
Right. But to find the bridge, they had to cross-reference that with osteosarcoma data.
So they have to look at the bone cancer side now.
Exactly. So they applied a statistical model called a lemma package to the bone cancer patient profiles.
And they're searching for what? Differentially expressed genes?
Yes, DEGs. These are genes that are drastically upregulated or downregulated compared to healthy bone tissue.
Okay, and that search yielded 563 genes.
Right.
So we have 1,410 suspects on the Li-Fraumeni side and 563 on the osteosarcoma side.
And when you mathematically overlay those two massive networks, you find an intersection.
A Venn diagram, basically.
Exactly, an intersection of exactly 51 shared genes.
Wow. 51 genes forming that crucial bridge.
But, from a pharmacological standpoint, 51 genes is still a haystack.
Yeah, you can't design a targeted therapy for 51 targets all at once.
No, you'd wreck the patient. They needed to isolate the absolute core drivers.
Which is where they brought in the heavy algorithmic artillery, right?
Yeah, they unleashed a battery of machine learning models.
And this is where the methodology gets really ruthless, because they didn't rely on human intuition here.
Right, they forced the AI to strip away the noise.
Yeah, they used 3 distinct algorithms. So first was random forest,
which essentially builds hundreds of chaotic decision trees.
Right.
Lets them all vote on which genes are most critical, and built a consensus.
Okay, then they applied lasso regression. And I always think of lasso like a ruthless corporate liquidator.
Oh, that's a great analogy.
Because it mathematically penalizes complexity, right?
Yeah.
It just shrinks the value of any non-essential gene down to absolute zero.
Leaving only the pure biological signal.
Right. And then finally, they ran SVM-RFE, which stands for Support Vector Machine Recursive Feature Elimination.
Yeah, and that one draws strict mathematical boundaries to eliminate the weakest variables recursively.
So it just keeps cutting the bottom performers until you're left with the best.
Exactly. And when the dust settled from that computational gauntlet, all three of those completely distinct algorithms agreed.
They all pointed to the same thing.
Yes, they agreed on just 4 core feature genes.
Okay, and those were LEPR, SOX18, TRIP13, and STIL.
Right.
But anytime you use machine learning in medicine, you run into the black box problem.
Oh, always.
Like the data goes in, the 4 genes come out, but how do we know we aren't just looking at a mathematical hallucination?
Well, you need transparency, right? Which is why the researchers applied SHAP values.
Oh, a Shepley additive explanation.
Yeah, which actually originates from cooperative game theory.
Which is so cool.
It is. It calculates and visualizes the exact marginal contribution of every single variable.
So it proves precisely how much blame or credit each of those 4 genes deserves in predicting the disease.
Exactly. And based on that transparent data, they built a predictive nomogram, which is a clinical scoring tool.
Okay.
And when they tested it, this nomogram achieved an area under the curve, an AUC accuracy score of 0.96.
0.96 out of 1.0.
Yeah.
I mean, in biological prediction, dealing with all the chaos of human genetics, that is a staggering level of precision.
It really is. But they still had to find the true ringleader among those 4.
Right, because there's still 4 of them.
Yeah. So by comparing the genes against real-world patient outcomes, the picture became crystal clear.
What did they find?
Well, LEPR expression actually decreased during metastasis. SOX18 and TRIP13, they didn't strongly correlate with overall patient survival.
But still did.
Yes. Still was significantly upregulated in metastatic tumors, and its presence was a direct harbinger of poor patient survival.
Wow. Okay, so let's pause and just anchor what we've uncovered so far in this first segment.
Good idea.
So we started with 2 massive genomic data sets. We found a 51 gene overlap between Li-Fraumeni and osteosarcoma.
Right.
Then we subjected those 51 suspects to a gauntlet of 3 distinct AI algorithms, whittling them down to just 4. And finally, clinical outcome data singled out STIL as the ultimate driver of metastasis and poor prognosis.
Exactly.
Which begs a fascinating question for you listening to consider. Imagine your genome has a built-in vulnerability like a TP53 mutation.
Right.
How does a secondary gene like STIL exploit that exact weakness to cause such chaos?
Well, to answer that, we have to zoom in even closer. The researchers had to abandon the macro view and descend onto the microscopic battlefield.
Right.
So they utilized single-cell RNA sequencing, or cRNA-seq.
And let's be really clear about why this technology is such a paradigm shift.
Oh, it changes everything.
Because in the past, analyzing a tumor was like, well, instead of blending the whole tumor into a smoothie to see what it's made of, single-cell RNA sequencing lets us taste every individual piece of fruit.
Yes, that resolution is key. It's like wiretapping every individual building in a city to hear exactly who is plotting what.
Exactly. So how many cells did they look at?
They analyzed an astounding 118,206 individual cells from osteosarcoma tissues.
That is massive.
It is. And they grouped them by their distinct genetic signatures, finding 13 distinct clusters.
Like immune cells, structural vascular cells, things like that.
Exactly. But they homed in on one highly malignant, highly active group.
The pro-OSCs, right? Proliferative osteosarcoma cells.
Yes. And to figure out what made these pro-OSCs so lethal, they analyzed them using the cytotrace algorithm.
Okay, cytotrace. What does that do?
Cytotrace measures stemness.
Ah, stemness. So in oncology, stemness is basically a measure of how youthful and unspecialized a cell is.
Right. How infinitely adaptable it is.
It's the engine of pluripotency, the ultimate chaotic potential for a cancer cell.
Exactly. And unsurprisingly, these pro-OSCs score the highest for stemness.
Makes sense.
Then the researchers mapped the cell's developmental timeline using what's called pseudo-time trajectory analysis.
Okay, so tracking the biological arrow of time.
Right. Reconstructing the tumor's life from its earliest inception to its mature state.
And what did they see?
They discovered that STIL is astronomically highly expressed at time 0.
Wow. So STIL is the initial spark driving that early chaotic proliferation.
Yes. What's fascinating here is that STIL doesn't just make the tumor grow. It's incredibly sinister.
Right, because it's not just accelerating growth, it's building a fortified bunker.
Exactly. It completely remodels the surrounding bone and immune environment.
It turns the cancer cell into this malicious architect, forcing it to communicate with its surrounding environment to build an immune desert.
Yes. STIL literally draws the map for the tumor's survival.
Which is actually a perfect transition. To help you remember how still high cells talk to their environment, think of the acronym MAPS. MAPS.
I love this. Let's break down that map.
Okay, starting with M.
M is for the MIF pathway. So STIL forces the tumor cell to pump out macrophage migration inhibitory factor.
So it's actively communicating with the surrounding immune cells.
Specifically, macrophages and B cells.
Wait, so it's not fighting the local immune system, it's essentially putting it on the payroll.
That's exactly what it's doing.
It's bribing the security forces to stand down.
Precisely.
Yeah.
It subverts the immune surveillance completely.
That is wild. Okay, so then we move to A.
Right, the APP pathway. Amyloid precursor protein?
Now, APP is something we typically associate with neurological pathology, like Alzheimer's, right?
Yes, normally. But here, STIL repurposes this neuron-like signaling, the APP-CD74 axis, to further suppress immune activity and protect that tumor microenvironment.
Just hijacking whatever tools it can find.
Exactly. Which brings us to P, the PTN pathway.
PTN standing for pleiotrophins.
Yes, and this is an autocrine loop.
So the cell essentially talks to itself.
Right. It secretes a chemical messenger that binds to receptors on its own surface via the PTN-NCL pathway.
Like a weird feedback loop to maintain its own youthful stemness?
Yes, preventing itself from maturing into a normal, harmless bone cell.
Okay, so that leaves S.
S is for space, facilitated by FN1 or fibronectin-1.
Because that addresses a massive mechanical problem for osteosarcoma, right?
A huge problem. A tumor needs physical room to expand, and bone is incredibly dense.
Right, you can't just push bone out of the way.
No. So STIL activates the FN1-CD44 pathway to signal osteoclasts.
And those are the specialized cells that naturally dissolve bone tissue.
Exactly. STIL sends them into overdrive, forcing them to eat away the surrounding bone to create a physical cavern for the tumor.
And the brilliant, terrifying bonus for the tumor here is that bone tissue acts like a vault for growth factors.
Oh, yes.
By forcing the osteoclasts to destroy the bone, STIL basically cracks the vault open, releasing all these trapped growth factors that feed the tumor even more.
It's a devastating mechanism. When you look at the MAPS pathways together, you realize STIL is functioning as a master coordinator.
Yeah, it's actively remodeling the structural bone environment and the immune environment simultaneously.
Right.
So, taking a step back and looking at this second phase of discovery, here's our mini recap.
Go for it.
We know STIL is most active in early, highly potent tumor cells, the pro-OSCs. We know it uses the MAPS pathways, MIF, APP, PTN, and space via FN1 to construct an immunosuppressive desert while destroying bone for expansion.
Perfectly summarized.
But knowing that this tumor has built a heavily defended communication network, it makes me wonder, how do we actually cut the phone lines?
That's the million-dollar question.
Because algorithms and single-cell sequencing are one thing, but how would you design a therapy to stop this? To answer that, we have to see what happens in a petri dish.
Right. To prove it in living biology, the researchers had to move to in vitro validation.
So transitioning from computer algorithms to physical cells.
Yes. They utilized 3 highly specific human osteosarcoma cell lines.
Okay, what were they?
First, SAOS2, which entirely lacks the TP53 gene. Second, U2OS, which possesses a normal, healthy, wild-type TP53 gene.
So that's the healthy baseline.
Right. And third, crucial for this study, the 143B line, which carries a mutated TP53 gene.
Ah, so the 143B line perfectly mimics the exact profile of a Li-Fraumeni syndrome patient.
Exactly. They needed to see what happens to the whole system if they surgically remove STIL from the equation in these different environments.
So how did they do that?
They used siRNA, small interfering RNA.
Which effectively acts like molecular scissors.
Yes, to silence or knock down the STIL gene.
And what happened?
The result was instantaneous. When STIL was silenced, the levels of the p53 protein shot up in both the wild type and the mutant cells.
Oh, wow.
This was a critical revelation. It physically proved that STIL acts as a direct negative regulator of p53.
So under standard physiological conditions, STIL actively suppresses the guardian of the genome.
Yes, it holds p53 back.
Okay, I have to challenge this logic for a second.
Sure.
Because if the p53 protein is already physically broken and useless in Li-Fraumeni patients, like we see in the 143B mutant cell line, why would the tumor spend energy keeping STIL around?
It seems like a waste of energy, right?
Yeah. If the brakes are already cut, why does the tumor care about a gene that suppresses the brakes? It seems totally redundant.
And that apparent redundancy is exactly what stumped researchers for a long time. It is the absolute crux of this paper.
Okay, so what's the answer?
The answer is that STIL's role is highly context-dependent.
What do you mean?
Well, when they knock down STIL in the normal, healthy U2OS cells, those cells just underwent cell cycle arrest.
They stopped dividing.
Right, because the normal p53 brakes were restored.
Makes sense.
But in the 143B mutant cells, the LFS mimic STIL's job entirely changes.
Wait, really? It's moonlighting?
Yes, it changes its career path based on the genetic neighborhood.
That is wild.
Since the p53 brake is already permanently broken, STIL abandons its role as a suppressor and becomes the primary engine driving invasion and metastasis.
Oh, wow. So how did they prove that?
They used a wound healing assay, where they literally scratch a layer of cells and observe how aggressively they migrate to close the gap.
Testing cell migration.
Right. And they also used a sphere formation assay, testing the cell's ability to form complex 3D structures.
Which is a pure test of stemness.
Exactly. And in the mutant background, knocking down STIL completely shattered the tumor's ability to migrate and maintain its malignant stemness.
Here's where it gets really interesting, though. We have mapped the mechanism, the environment, and the context dependency.
Right.
But how do we weaponize this biological insight for the patients actually fighting this disease?
This is where we bridge biology to pharmacology.
Okay, clinical application.
Yes. The researchers analyzed drug sensitivities and discovered that tumors with high STIL expression possess a massive Achilles heel.
What is it?
They are exquisitely sensitive to a class of drugs known as WEE1 inhibitors.
WE1 inhibitors.
Yes.
Okay, so to understand why this is such a lethal vulnerability, we have to look at the cell division cycle, right?
Exactly. Because a cell doesn't just divide recklessly.
Right. It has to pass through security checkpoints to make sure its DNA isn't heavily damaged.
Yes. Normal cells have two primary checkpoints. The first is the G1S checkpoint, which is heavily regulated by p53.
Okay.
The second is the G2M checkpoint, which occurs right before the cell physically splits. And this one is controlled by an enzyme called WEE1.
But in Li-Fraumeni syndrome, that first checkpoint is completely obliterated because of the TP53 mutation.
Exactly. The tumor cells are blazing right past security with heavily damaged mutated DNA.
Which means those cancer cells are surviving on a tightrope.
Precisely. Because they bypass the first checkpoint, they rely entirely, 100%, on that second WE1 regulated checkpoint.
They need it to frantically patch up their damaged DNA before they undergo division.
Yes. So if you administer a WE1 inhibitor,
you kick out their only remaining crutch.
You block WEE1, and the cancer cells are forced into division with massively shattered, unrepaired DNA.
Which triggers a phenomenon biologists call mitotic catastrophe.
Right. The cells don't just stop growing. The physical act of division fatally tears them apart.
It's using the cancer's own chaotic momentum against it.
It's brilliant. And to underline just how monumental this vulnerability is, the researchers didn't stop at osteosarcoma.
They ran a sweeping pan-cancer analysis, didn't they?
They did. And they discovered that STIL overexpression isn't just an anomaly in bone cancer.
It's everywhere.
It is a major active threat driving poor prognosis in 28 out of 33 different human cancer types.
28 out of 33.
Yes. It represents a near universal vulnerability being exploited by malignancies across the entire human genomic spectrum.
Okay, so let's synthesize everything we've unpacked today.
Sounds good.
So we started with a terrifying clinical mystery from 1969, a broken TP53 gene causing aggressive bone cancer in LFS families.
Right.
We discovered that the pathway from mutation to tumor is not a simple linear chain of events. STIL actively suppresses p53, but its role is context-dependent.
Exactly. In a healthy cell, it stops STIL division.
But when the p53 guardian falls, STIL senses the context shift. It becomes the architect of chaos, exploiting the missing breaks to drive metastasis.
Remodeling the tumor's physical space, immune environment, and cellular stemness.
But in doing so, it leaves the tumor entirely dependent on the WEE1 checkpoint, which opens the door for mitotic catastrophe. WE1 inhibitors represent a massive vulnerability for these tumors.
Yes, and this completely shatters the old paradigm of single mutation oncology.
Right.
It proves we can't just study a mutation in a vacuum. We have to interrogate the entire genetic neighborhood to see who steps up when the primary systems fail.
So what does this all mean for you listening? If a gene like STIL completely changes its job description based on the background mutations of the cell, how does this change the way we think about universal one-size-fits-all cancer drugs?
It challenges the entire premise.
It really does, and I want to leave you with a final thought to mull over or explore on your own.
Oh.
Since that pan-cancer analysis revealed STIL is manipulating outcomes in almost all human cancers, it raises a profound question.
Which is?
How many other secondary genes are out there right now, quietly pulling the strings in the shadows of famous headline-grabbing mutations like TP53?
That is a chilling thought.
Right. For decades, the medical field has been staring intently at the broken locks. Is it possible we've been completely ignoring the thieves casually walking through the front door?
We might just be scratching the surface.
Well, thank you for joining us on this intensely detailed journey into the multi-omics landscape. Keep questioning the complex systems around you, and we'll see you next time.